नमस्ते स्टूडेंट्स, खुश रहो बेटियां। I, JV and Dr. Y. Chandrakala, Assistant Professor, Vidyapeeth, Women's University, Jaipur. Okay. So, myxomycetes, a true or a plasmodial slime mold. Myxomycetes are the largest group of the slime mold and comprise of 800 species and 62 genera. Okay. These slime molds are common in moist and decaying wood and other organic substances. They are also abundant in the soils and many fulfill the ecological functions. Okay. So the vegetative phase is a free-living plasmodium, that is the multinucleate or wall-less mass of the protoplasm. And this may be covered with a thin sheet of slime sheet. Okay. Plasmodia vary in size and can be loosely grouped into three categories: protoplasmodia and aplanoplasmodia. Protoplasmodia in the microscopic structures usually give rise to the single sporangium, and they resembles into the simple plasmodium or protosteliate. Plasmodia is the thin open network of the plasmodium strands and are transparent in the individuals 5 to 10 micrometer. Sternoplasmodia is the large sheets or a network with with veins, and within them the protoplasm shows rhythmic and reversible streaming and pulse lasting of 60 to 90 seconds. This striking phenomena is readily observed with a dissection microscopes and probably due to the interaction of calcium ions within the skeletal element lining of the veins. Okay. So the life cycle of the myxomycetes is a typical, like it can be like the plasmodium is diploid and feed by the phagocytosis of the bacteria, yeast and fungal mycelia, or a spore. It can raise to the sporophores under the appropriate conditions. The haploid spores are dispersed. So here you can see in the life cycle of the myxomycetes here. Okay. So the spore released from the sporangium are haploids. Okay. And can germinate by releasing either the single amoeba or swarmers. Okay. These are the swarmers. And these two cell types are convertible. Okay. The maxoamoebas can divide mitotically and okay, usually take place between the swarmers, and which must be belonging to the different mating types. The karyogamy K follows the diploid zygote, establishing the pheneroplasmodium. Okay. When the nutrient become limiting and the sporophores is formed and differentiated into the sporangia, in which the meiosis occur. Unfavorable conditions can be overcome at a haploid stage. Okay. So when the maxoamoeba forms the microcyst at the diploid stage, when the plasmodium forms the sclerotic or open or closed circles, okay, repeatedly forming the nucleus and opposing the mating type, and diploid nucleus are large and half filled. Okay. So here it depends upon the wind conditions and such as the moisture, germination and releasing of each of the amoeba or the zoospore or the swarmer with usually two anterior whiplash or the flagella. Which one is shorter than the other and thus forming an invisible. The amoeba are called the myxomycetes and if the order distinguish them from the amoeba and of the cellular slime mold, which have a different functions in the life cycle. Okay? Myxomycetes are capable of asexual reproduction by division. Okay, asexual reproduction by the division. Okay, swarmers cannot divide, but can readily or reversibly convert to myxomycetes and secretes a wall to form a microcyst. Both swimmer and the amoeba, maxamoeba forms a pillows pseudopodia with the help of the and engulf their prey. So the sexual reproduction is initiated when the two haploid maxamoeba are or the swimmer of the compatible mating types fuse to form a zygote, from which the diploid plasmodium develops. And the plasmodium can survive or adverse condition by turning into a resistant sclerotium in which numerous walled compartments or each containing the several nuclei are formed. Okay. So here the depend upon the species, the sporophores may be take place in the shapes and intermediate between and thus different type of sporophores are possible. Okay. The plasmodium can be maintained sterile or plasmodium can be maintained for a prolonged period in a vegetative state and the sclerotic can be stored in a dry for months. Okay. The spore have been retrieved after like 50 years also in a storage in a herbarium. Thank you. This session is powered by 2.0 Jyoti Vidyapeeth Women's University, Jaipur. If you have any queries, we'll discuss in the problem solving session. Thank you.